Hello, it's me again, Noam Levaot from Ben-Gurion University. I would like to start from thanking the organizer, letting me speak in this workshop and even speak twice. I hope you will be interested. So David Karasik, thank you. You gave me a lot of work, but I enjoy it and I appreciate it a lot. So I'm going to talk today about engineering proteins for the treatment of patients with bone diseases. And it is already well known that osteoclast and osteoblast talk with each other. There is coupling of their activities, and osteoclast actually signals to osteoblast while they resorb and encourage osteoblast proliferation of progenitors, differentiation, and function. And more coupling factors are being discovered. And the proof for this coupling and the potential of this coupling as a therapeutic strategy was emphasized by the drug odonakitib, which targeted cathepsin K. And cathepsin K inhibited osteoclast resorption. It didn't inhibit the differentiation. Osteoclasts were generated, they sat on the bone, but they couldn't efficiently resorb the bone, but still signal to osteoblast, and the result was very promising, although side effect has been ruled and canceled. This drug was did not reach the clinic. But even before odonakitib, we already have seen some clues for osteoclast resorption dysfunction that encourage bone formation. And what caught our attention is a paper published by in the group of Stephen Teitelbaum in 2000, and he has found that mice deficient in beta-3 integrin are osteosclerotic, and they have a lot of osteoclasts, and osteoclasts do not resorb well the bone in these mice, but still signal, and there is increased bone formation in these mice, and they have sclerotic bone. But they also discovered that this was transient. So the osteosclerosis was apparent only when the skeleton matured, and afterwards bone was resorbed and they had normal bone mass. So they concluded from there that the high amount of osteoclasts generated in these mice compensated for the lack of their activity. And they also found the machinery which they published in a later publication, showing that these mice had very high level of M-CSF, macrophage colony stimulating factor. It's a cytokine. And when they culture beta-3 integrin with high amount of M-CSF, they saw more mature osteoclast forming. This also slightly increased the level of their resorption. So they concluded that M-CSF can compensate or can help osteoclasts that don't have beta-3 integrin. And finally, a later publication showed that CFMS, the receptor for M-CSF, collaborate with alpha V beta 3. CFMS is a growth factor receptor, and the two receptors form complexes, and we know that M-CSF regulate these complexes, and a high dose of M-CSF can rescue the osteoclastogenesis of beta-3 deficient osteoclast. So this close collaboration between the growth factor receptor and the integrin receptor was the rationale for us to try to design and engineer an inhibitor that would inhibit simultaneously, bind and inhibit simultaneously both receptor activity in osteoclast. So in order to do that, we first try to inhibit or design an inhibitor for CFMS. CFMS is a tyrosine kinase receptor. It's autophosphorylate itself, and it does so when it forms dimers. And what induces the formation of the dimer is a dimer of M-CSF molecules, which you see here. So what we did, we mutated the binding, the dimerization site in M-CSF, and therefore we generated an M-CSF monomer. So when a monomer bind the receptor, it cannot bring two receptors together and there won't be any signaling. The other thing that we did is we choose to target one of the, or two of the loops of M-CSF and introduce an RGD motif there. RGD bind integrins in order to bind the alpha V beta 3 integrin, but RGDs alone do not bind integrins. They need the flanking amino acid, and the flanking amino acid of the RGD integrin substrate determine which receptor they are going to bind. So what we did, we used the library with random mutation around the RGD, an RGD motif in the two loops of M-CSF to select for M-CSF that can bind alpha V beta 3 integrin. And in order to do this, we used a very powerful system named yeast display, in which you display your protein of choice on the membrane, on the cell wall of membrane of yeast, and this target protein, in this case was our M-CSF library, has a tag that can be identified by a protein, so we can get a signal by just expressing this protein. And then we use soluble target protein, in this case our receptor, alpha V beta 3 and CFMS, in order to determine how which cells or which proteins or mutants in the library bind these proteins the best. And the way you do it is by enrichment of the library through sorting. So we sorted cells, yeast cells that express the M-CSF, you can see it's on the right half, and that bind the specific protein. And what we did is we took the M-CSF, we knew it bind CFMS with high affinity, and then we started to select for alpha V beta 3 binders, and each time we reduced the level of alpha V, the concentration of alpha V beta 3. So the idea is to select, reduce the concentration, sort for the binder, then enrich the binder, select again and again and again until you reach the nanomolar levels. And at the final stage, we selected again for CFMS to see that this group didn't lose their binding affinity for CFMS. So after isolating a few clones, we purify the proteins in yeast, and we perform SPR analysis to determine their affinity. And what you can see here when we look at the double binders is that they can bind both receptor with high affinity. So if we use a monomer that doesn't have the RGD motif, it cannot bind the alpha V beta 3 integrin. We could select for alpha V beta 3 for M-CSF that were mutated in their ability to bind CFMS, and then we got only selective binding to alpha V. And when we use the B specifics, we got binding to both proteins. We isolated 3 types. We can see that we gave them number according to their clones, and here you can see their affinities, and we got affinities in the nanomolar range for CFMS and alpha V beta 3. The star of this talk is going to be the one that is named 5.6, which had a very high affinity to CFMS and a lower affinity to the alpha V beta 3. So after doing all these artificial assay, we asked if these can really bind cells. and we use cells that express both alpha V beta 3 and CFMS, and we culture them with the inhibitor. So, and this time we use our protein, the inhibitor, is 5.6, which was conjugated to a fluorescent tag. So it has a fluorescent, and this was our reference. And then we use the same concentration of the protein, but this time with a competition of untagged wild type M-CSF. So we could see that there was a competition, and the wild type competed with our protein on binding to the cells. The other thing we did is to do the same assay with an RGD, cyclic RGD, untagged cyclic RGD, and we got also reduction on binding, meaning that both M-CSF wild type and cyclic RGD, the ligand for integrin, the ligand for CFMS and integrin, compete with our protein on binding to cells, suggesting it binds the same receptors on the cells. Interestingly, when we use the combination of both, we get the greatest competition. So suggesting that our protein binds both of the receptors, and in order to eliminate its binding to the cell, you need to compete on binding of both of the receptors. As expected, the monospecific that can protein that can bind only the CFMS receptor or the alpha V beta 3 could not achieve this level of competition. So the next thing was to ask if we really inhibit the signaling cascades that are regulated by alpha V beta 3 and CFMS. And here you can look at the CFMS phosphorylation, autophosphorylation. This is the initial step in CFMS signaling. So this is the phosphorylation of positive control cultured with M-CSF. This is without the M-CSF, so there is no phosphorylation. And this is when we put M-CSF together with any of the clones that the B-specific clones, we see a nice reduction, a significant reduction in CFMS phosphorylation, suggesting we inhibit this pathway. To look at the alpha V beta 3 integrin, we didn't look at signaling cascade, we looked at the osteoclast cytoskeleton. And here you can see osteoclast in culture stained for the actin cytoskeleton. The typical osteoclast form these sharp actin bands in its periphery. And and this is dependent on intact alpha V beta 3. If you use a cyclic RGD that disturb beta 3 activity, you can see that the sharp edges of the actin belt are gone, and we see something like a blue appearance. When we used any of our inhibitor, we could see the same effect. It was very strong, even with 422. That was the best integrin binding. So that was really supporting our hypothesis that we are inhibiting integrin beta 3 here. And when we use the monomer that only targets CFMS, we could see an effect, but it was much less intensive than what we see when we inhibited the integrin directly or used the B-specific inhibitors. So after doing this, we wanted to see what are the effects on differentiation. And when we use concentration as low as 50 nanomolar, we could see a nice inhibition of osteoclast differentiation. So osteoclasts, these are trap-stained osteoclasts in culture. They form these large multinucleated cells. They need M-CSF and rank ligand to do that. If you remove rank ligand, that's what you get. When we culture with both of these cytokines and together with elevated concentration of the inhibitors, we could see nice inhibition of the osteoclast formation in vitro. We did it both on mouse cells and on human cells, and we saw in mouse a very nice dose-dependent effect of the inhibitors. Look at the osteoclast number here. We saw the same effect on human. That was a significant inhibition. The best dose-dependent effect we saw with the 5.6, and this was one of the reasons we chose to continue with this protein. This is just to show you the purification and isolation of the M-CSF protein, and the size here is big, and the reason that the size here is big is that M-CSF is small, and if you inject it, it would go in the urine very fast. It would be filtered out, so we increased the protein size by attaching human serum albumin to the protein. So we had a chimera protein, which was a M-CSF, monomeric M-CSF fused to HSA, and we named it 5.6 HSA. So that's what you're going to see from now on. So we injected this protein in vivo to mice through the performing subcutaneous injection, and we wanted to ask where if the proteins, if our B-specific protein goes specifically to bones, or it's getting stuck in some other tissue. So we tagged again this protein with the fluorescent marker and looked at its distribution using IVIS in vivo. So here you can see all kind of organs, heart, lungs, etc. There is no signal in any of these organs. Our control was uninjected mouse, a mouse that was injected only with the dye, so there was no signal with the dye. A mouse that was injected with M-CSF without HSA. So, and this is 1 week after injection. It's important, I forgot to tell you. And what you can see here below are bones. And in order to be able to see what's going on inside the bones, we cut them open longitudinally and cut the epiphysis. So you can see the epiphysis and you can see parts of the diaphysis of this bone. And we saw a very nice signal that was detected only here in bones that had the 5.6 HSA 1 week after a single injection. So it goes directly to bone, it's accumulating there, and it doesn't accumulate in any other organ. We also monitor the level of the protein in the blood. So after subcutaneous injection, you have a sharp increase of the protein level in the blood. And during 100 hours, we can't detect it any more. So after doing all this pharmacokinetics experiment, we went to perform an experiment in mice. We chose the ovareectomy model. So we either ovareectomy or sham-operated mouse, let them recover for 7 days. And after 7 days, we injected subcutaneous the 5.6 B-specific protein. We also used alendronate as a positive control for treatment. And we used a protein that was one of the monospecific that could bind only CFMS, but not alpha V beta 3. The experiment was 28 days. Then we sacrificed the mouse. We did calcium injection before that. And the tissues were analyzed blood was analyzed by bone turnover markers. Bones were analyzed by histology and micro-CT. So from here, we saw very nice result when we looked at biomarkers. So this is CTX1, the marker for bone resorption. This is a cross-link collagen that is traveling in the blood and you can detect by ELISA. So these are the ovareectomized mice. We have high level of CTX in their blood compared to the sham-operated. When we compare the injected mice, the treated mice, we see that they had also significant reduction in CTX levels compared to the ovareectomized mice. And pay attention, we see here 10 milligrams. So we used 3 different concentrations, 10 milligram per kilogram, 2.5 milligram per kilogram, and 1 milligram per kilogram. I will tell you later why this is important, but you remember here, so at least from our cohort, we see a significant effect on the high concentration. The middle concentration, 2.5, doesn't seem to give significant, at least on this cohort, 1 milligram the same. The monospecific was also not significant, and the alendronate, of course, does its job. It reduces CTX levels. So here it becomes interesting. We look at bone formation marker. So what happens when you overreactomize mice, CTX levels goes up, but bone anabolic function also goes up at the first period of time, but still resorption is higher than formation and mice lose bone. So you can see it here. This is the overreactomized. They have higher level of PINP, the bone formation markers, compared to the sham operated. When we inject 10 milligrams per kilogram, the highest concentration significantly increase bone formation in these mice, in the level somewhere at the level of the overreactomized one. We see the same effect even with the lower concentration. When we use 2.5 milligram and we saw the same effect. You can even see these mice that had super high PINP levels at the lower concentration of our protein. When we look at the RDG, the 10 milligram per kilogram, the one that can bind only CFMS, had also significant increase in bone formation. Remember, this one did not give us any significant reduction in bone resorption. And the alendronate was very low. So alendronate was significantly lower than the overreactomized, and this is known. So one of the problems of current resorptive drugs is that they completely shut off bone formation. So we looked at bone volume fraction in the vertebra of these mice. So this is the overreactomized. We see a sharp reduction compared to the sham operation. They lose a lot of bone. We had the same effect when we used 10 milligram per kilogram. We had completely blunted bone loss by the overreactomy. And when we used the 2.5 milligram per kilogram, and that was the most surprising result, we got a very high bone mass compared to the sham operated, even the healthy mouse, and of course, much more than the 10 milligram per kilogram. So what's going on here? When we use the HSA, the lower concept, 1 milligram per kilogram, that was probably too low. We got the same effect as the 10 milligram per kilogram. The monospecific didn't have a significant effect on bone mass, and alendronate had increased bone mass, also had a significant effect on bone mass, shuttling off completely resorption. So when we look at the bone, and we looked at what I'm going to show you is trabecular number and trabecular thickness. Trabecular number is a parameter that is associated with both bone resorption and bone formation. And you can see much higher level of trabecular number in the sham operated compared to the overreactomy mice, the osteoporotic mice. We could rescue the loss of trabecular number when we use 10 milligram per kilogram. We had even a better effect, again, with the lower concentration. And even going to 1 milligram per kilogram. When we use the monospecific, that didn't reach significance in this cohort, and the alendronate gave a significant increase in trabecular number. When we look at trabecular thickness, which is more associated with bone formation, our highest concentration was not significant in increasing trabecular thickness, with a very significant result with the 2.5 milligram, the middle concentration, and the 1 milligram per kilogram also was significant. At this case, targeting only CFMS gave a positive result. It wasn't as the same as the lowest concentration, and the lendronate was also positive. So why I'm telling you all this? Because M-CSF is actually acting at two different stages during osteoclast differentiation. We know that we need M-CSF in order to direct monocyte into the osteoclast fate, and it's involved in the differentiation of the cells. So using higher concentration of RB specifically, what we think was probably inhibiting osteoclast differentiation. And this is the reason that we saw a significant reduction in resorption, but because you inhibit only osteoclast differentiation, we also did not have a significant, or our effect on bone formation was lower, and when we use the 2.5 milligram, when we use the low concentration, we don't see such an effect on bone resorption, but we have increased bone formation because the osteoclast sits there. It doesn't produce, it doesn't resorb well, but it produces the coupling factor, which encourage formation. So we hope that this approach, this strategy, could actually produce a bone resorption compound inhibitor, but that may facilitate, may allow bone formation to go on, and even in certain concentration, encourage bone formation. And that would be different from all the resorption, the bone resorption compounds that are today in the clinic. So I would like to finish, first of all, with thanking Dr. Yuval Tsur. This was a former PhD student. He did most of this work. He's now in the States in a biotech company in San Diego. And of course, my lab manager, Dr. Chendan Abramovich, and PhD student Sveta Kachkovsky, which also helped, and we'll talk about her project in this meeting, and my very close collaboration and fruitful collaboration with Dr. Niv Papo here in Ben Gurion University. Thank you all for listening. I hope you enjoy it, and see you.